Hello everyone. Now we will discuss on the topic instrumental techniques for environmental analysis part 2. In the part 1 of this topic we have discussed on gas chromatography and liquid chromatography, and this class we will discuss on the spectroscopic analysis, that is UV spectrophotometer and FTIR. Now we will see first UV visible spectrometry. So, spectroscopy is the study of the properties of matter through its interaction with various types of radiation, normally electromagnetic radiation of the electromagnetic spectrum, and spectrometric techniques are a large group of analytical methods that are based on atomic and molecular spectroscopy. Spectrometric methods refer to the measurement of the intensity of radiation with a photoelectric transducer or other types of electronic device. And UV visible spectrometry is one of the oldest instrumental techniques of analysis and is the basis for a number of ideal methods for determination of micro and semi micro quantities of analytes in a sample. It concerns with the measurement of the consequences of interactions of electromagnetic radiation in the UV and or visible region with the absorbing species like atoms, molecules or ions. That means, if we have one sample and then if we radiate some electromagnetic radiation like say light, so then it will be capturing some part of it and different elements will be capturing different light or radiation of different wavelength, and the intensity of the radiation will also be reduced when it will pass through the sample. So, by decrease in the intensity we can quantify, and by knowing the wavelength of the radiations that is being absorbed by the molecules or the element, we can know what type of molecule it is or the element it is. So, it is an analytical technique that measures the amount of discrete wavelengths of UV or visible light that are absorbed by or transmitted through a sample in comparison to a reference or blank sample. When ultraviolet radiation are absorbed, this results in the excitations of the electrons from the ground state towards a higher energy state. So, when we are sending some radiation like say UV radiation or visible also, UV and visible, UV is more powerful than the visible, then both are having some capacity to excite the external electron from their base to the excited state. For example, here you see these are base position and these are excited position. So, electrons may transfer from base to excited state, and different types of electron can be available like say pi electron or non-bonding electrons or n electrons. Okay. So, they can absorb energy in the form of ultraviolet light to excite these electrons to higher anti-bonding molecular orbitals. The more easily excited the electrons, the longer the wavelength of the light. The longer wavelength means less powerful. Okay. They can absorb. There are 4 possible types of transmission, that is pi pi transmission, n pi transmission, sigma sigma and n sigma transmission. That means, these 4, as it mentioned here, 4 types of electron that can be shifted to 4 other positions, and they can be ordered as follows, that is this is greater than n sigma and pi pi and n pi. So, these are there, and the absorptions of ultraviolet light by a chemical compound will produce a distinct spectrum that aids in the identifications of the compound. So, how the UV visible spectroscopy or spectrometry works? It works on the principle of Beer-Lambert law. What is that principle? So, if we pass some radiation or UV light through a sample which is present here. So, when it will be passing through it, the some part of light or the radiations will be electromagnetic radiations will be absorbed, and particularly in this case of UV light will be absorbed and the intensity will be reduced. So, here we have having more intensity, here we will be having less intensity, and that log of I0 by I, that is proportional to length of this and concentration of the element present in it. So, that is equal to log I0 by I is equal to some constant into L into C, where E is equal to nothing but the molar absorptivity, and C is the concentration of sample and L is the path length of the sample, and this molar absorptivity E is equal to A by C L, and A is the absorbance. So, this is the relationship. So, we can get it here E is equal to log I0 by I divided by L C. So, from this expression we can get is equal to log I0 by I divided by L into C. Now, this is nothing but absorbance A. So, this is our absorbance A. So, absorbance, it is reciprocal of the transmittance also. So, we will see in reciprocal of the transmissions. Okay, so absorbance A is the optical density and log I0 by I. So, absorbance is also called as optical density. So, now you see, so what will happen, more the concentration, so this value will be more, because I value will be less than the I0. So, this ratio will be more. So, this absorbance value will also be more. So, more the concentration, we will be getting more absorbance value and less intensity of the transmitted light. So, now if we can have some detector, so that will detect the quantity of light which is being transmitted. So, that can give us some idea the how much is light is absorbed and which is proportional to the concentration present in it, and we can get the value of the concentration of the element. Now, if we see the different parts of the UV visible, then one is that we have to send some light, UV light. So, light source is needed. So, tungsten filament lamps and hydrogen deuterium lamps are the most widely used, and we have to send a monochromator. Okay. So, monochromators generally are composed of prisms and slits. We have to send one wavelength only, one ray, one type of ray or wavelength we have to send. So, most of the spectrophotometers are double beam spectrophotometers, and the radiation emitted from the primary source is dispersed with the help of rotating prisms. The various wavelengths of the light source which are separated by the prism are then selected by the slits, such the rotation of the prism results in a series of continuously increasing wavelengths to pass through the slits for recording purposes. The beam selected by the slit is monochromatic and further divided into two beams with the help of another prism. So, this is the how the monochromatic light is generated by the use of this monochromator. Then we will be using one sample and another reference. So, through both the light will be passed and we will be measuring how much light is being absorbed. So, that will be your sample and reference cells, 2 cells are needed, and if it is a double beam, then simultaneously the light 2 beams will be sent, one through the sample and another through the reference, and if it is a single beam. So, it will be sent once, it will be sent to the sample, and after that it will be sent to the reference, not simultaneously. So, that way 2 different arrangements are there, and then after that we will detector is necessary. So, generally 2 photo cells serve the purpose of the detector in UV spectroscopy. One of the photo cells receives the beam from the sample cell and the second detector receives the beam from the reference. The intensity of the radiation from the reference cell is stronger than the beam of the sample cell. This results in the generations of pulsating or alternating currents in the photocells. So, photocells, the intensity is changing and the intensity of light which is emitted from the, which is transmitted from the sample is lower than that of the reference. So, there will be some variations and pulsating or alternating currents will be generated, and then that amount of current which is generated, that is not very high. So, that has to be amplified. So, amplifiers are used. The alternating current generated in the photocells is transferred to the amplifier. The amplifier is coupled to a small servometer, and generally the current generated in the photocells is of very low intensity. The main purpose of the amplifier is to amplify the signals many times. So, we can get clear and recordable signal. So, recording devices are also needed, and most of the time amplifier is coupled to a pen recorder which is connected to the computer, and the computer stores all the data generated and produces the spectrum of the desired compound. So, this is the working of the UV spectrometry. So, here is the light source. So, wavelength selectors, that we are having monochromator, that is monochromatic light we need to produce. Then shutters are there. So, these shutters will allow the passage of this light. So, here it is going there. So, this is reference, other time it will be passing through the sample, not in the same time, because it is single beam, because from this we are getting one beam only. So, then it will be going to the photo detector, and for photo detector the signal will be formed and that will be amplified, and then it will be going for output device, and this is for double beam spectrometers. So, in this case source, wavelength selector is giving us 2 beam, these 2 beams. So, these 2 beams are simultaneously, it is one beam is going through the reference and another is going through the sample. Then this is we have photo detector, here also photo detector. So, then the difference is giving some signal and that is amplified, and again it is going to output device. So, this way the UV works, UV visible spectrometry works, and double beam or single beam spectrophotometers are available. So, UV and visible, the main difference is their wave number. The monochromatic light which we are generating, that wave number, the visible is having more wave number, wavelength as you mentioned here. So, the ultraviolet region corresponds to 400 to 500 nanometer and visible region, so 400 to 800 nanometer. This is your wavelength. Now, detection of impurities. Okay, it is one of the best method for the determination of impurities in organic molecules, and additional peaks can be observed due to impurities in the sample, and it can be compared with that of the standard raw material, and by measuring the absorbance at a specific wavelength, impurities can be detected. So, particular type of compound or element, those will be able to capture or absorb light of that particular wavelength. okay, or monochromatic light of a specific wavelength. So, if we see our major light intensity we are getting for a particular wavelength, but minor absorptions we are getting in other wavelength, that means that minor is basically because of the impurities. And UV absorption spectroscopy can characterize those types of compounds that absorb UV radiation, thus used in the quantitative determination of compounds. Identification is done by comparing the absorption spectrum with the spectra of known compounds. Like other methods, here also we need some standard molecules, and kinetics of reactions can also be studied using UV spectroscopy. The UV radiation is passed through the reaction cell and the absorbance changes can be observed. So, that way we can measure how with time the concentration of the element or the pollutant is changed. So, UV visible has some advantage as well as disadvantage. So, advantages are, the core advantage is the accuracy of the UV visible spectrophotometer, provide robust operation. UV visible spectroscopy is simple to operate and cost effective instrument, and cover the entire of ultraviolet and visible range. So, wide range of wavelength they can handle, and it can be utilized in the quantitative and qualitative analysis, and it has many disadvantages as well. The results of the absorption can be affected by pH, temperature, contaminants and impurities, and only liquid samples are possible to analyze, and it takes time to get ready to use it, and cuvette handling can affect the reading of the sample and may not be suitable for analysis in trace concentrations. The instrument has some detection limit. So, detection limit for UV visible may not be very less. Okay, if it is ppb, then it may not be able to give us. Then next is FTIR spectrometry, that is Fourier transform infrared spectrometry, and FTIR stands for Fourier transform infrared. The preferred method of infrared spectroscopy. In infrared spectroscopy, IR radiation is passed to the sample, unlike UV visible. UV visible, in case of UV visible we pass UV visible range of monochromatic light, but here in FTIR we will be passing IR light. So, IR we know the wavelength is more than that of the UV visible, more wavelength, and so less powerful. So, the IR will not be able to shift the outer shell electrons from its base to an excited state, but it can give some vibrations or rotations of different bonds. So, FTIR gives us more idea about the functional groups present in the molecules, etc. So, the resulting spectrum represents the molecular absorptions and transmission, creating a molecular fingerprint of the sample. Like a fingerprint, two unique molecular structures cannot produce the same infrared spectrum. So, this makes infrared spectroscopy useful for several types of analysis. These absorptions corresponds specifically to the bonds present in the molecule. The frequency range are measured as wave number, typically over the range 4000 to 400 centimeter inverse. So, you see here, this is the FTIR spectrometry. We have one IR source, then we have the sample. So, when the light will pass through it, then it will be, some will be absorbed and rest will be going out, and then we will be detect it, and we will see at which wavelength the light is available here. So, information provided by FTIR, it can identify unknown materials, it can determine the quality or consistency of a sample, it can determine the number of components in a mixture, and in this case So, we have a light source. So, this light is coming to this mirror, there is a number of combination of mirrors. So, that is called interferometer. So, interferometer is the most important part of the FTIR spectrometry. So, here it is coming to this mirror, and then from the parallel beams are coming, it is through this one, that is your beam splitter, and then through the beam splitter it some light passes through it and some is your reflected, and there is one movable mirror and another is your fixed mirror. So, again these two mirrors reflects the light this direction. So, one it is coming and again it is coming to this, and again it is one part is coming here, the that part is another, it is after reflection it is going there also. So, and these two two beams are merging here and going as a single beam. So, this single beam is passed through this sample chamber. So, here we will put the sample and then it will be detected, what type of our detector will be able to detect this one. So, from the sample this ray will be going out and it will be coming to the detector. So, that way. So, the typical FTIR spectrometer consists of an IR light source, interferometer, sample compartment, detector, amplifier and computer, and the light source generates radiations which strikes the sample passing through the interferometer and reaches the detector. Then the signal is amplified and converted to digital signal by the amplifier and analog to digital converter respectively, and eventually the interferogram is translated to spectrum through the fast Fourier transform algorithm. So, that is it is called FTIR, and the interferometer is the main core of FTIR spectrophotometer or spectrometer. The interferometer consists of a beam splitter, fixed mirror and a movable mirror that translates back and forth very preciously, that just we have discussed, and the beam splitter is made of a special material that transmit half of the radiation striking it and reflects the rest half of the radiation. It works on the basis of the principle that the light from the source is collected by collimating mirror, just like this collimating mirror we have discussed, and made its rays parallel, which strikes beam splitter and consequently splits into two beams. So, this is the mechanism. One beam is transmitted through the beam splitter to the fixed mirror and the second is deflected off and the beam splitter to the moving mirror, and the fixed and the moving mirrors reflect the radiation back to the beam splitter, and accordingly both of these reflected radiations are recombined at the beam splitter, resulting in one beam that leaves the interferometer and interacts with the sample and strikes the detector. So, principally the FTIR spectrometer collects and digitizes the interferogram, performs the FT function and displays the spectrum. So, this is the role of the FTIR, just like this source, then interferometer, then it is going to the sample, the single beam, and then detector, then interferogram which are getting, that is converted to the spectrum. So, that is why FTIR. So, here we are having transmissions and then transmittance, and this is our wave number. So, if we see this type of spectra we will get. So, this is our transmittance, percentage transmittance, and this is our wave number. So, in this wave number we see we get different peak here due to transmittance. So, these two, this range it is CH stretch, OH stretching, this to this 3200 to 3400. Similarly, 2100 to 2260, CN or C triple C, CC triple bond. So, these are the different functional group as we are talking about. So, now if C double bond strength is responsible for this peak. Okay. So, and in this range say 500 to 1000, this is basically fingerprint region, and this is not of that useful for organic compounds. This is another example. Here also single bond stretching, triple bonds, double bond. Okay. So, as the IR has low energy, so as I have mentioned, it is absorbed by the molecules as per the different vibrations and rotations of the double and triple bond. So, that way we can, the different peaks are generated. So, this peak is responsible for CO, CC, CN, CO, CN, CC like this. So, nitrile, if it is nitriles or carbines, so within this range we can get some peak. So, there are some library also. So, that gives that which at which wave number what type of compounds or what type of functional groups can be responsible, okay, or can be available. Now, how to put the sample in case of FTIR? So, we will see the FTIR can analyze solid, liquid and gas samples, and if it is a solid sample. So, there are some defined method which has to be followed to use it for analysis, like say NaCl plates can be used or potassium bromide can be used for solid samples. So, if NaCl plates are used, in that case, so the technique is called pressed plate, and when KBR the technique is thin film. So, in this, in the first case for NaCl plates, the sample is dissolved in a solvent which is non-aqueous and non-reacting, non-IR absorbing in the range to be measured. So, this is the property of the adsorbent, and a drop of this solution is placed on NaCl plate and the solvent evaporated to dryness, leaving a thin sample film. So, this is one prescribed method we need to follow for the analysis, and when we will go for KBR method. So, then 1 milligram sample to 100 milligram of KBR is finely ground and compressed into a transparent disc using an evaluable dye and hydraulic press. So, the solid sample may have some color also, and it may not be that transparent. So, to increase its transparency, this KBR is used, and the sample is liquid, then NaCl liquid cell can be used, and this method is neat sample, and the cell consists of two high quality pressed NaCl windows sealed and separated by a thin Teflon gasket and weighted with mercury. Shell thickness for most liquid is usually 0.01 to 0.5 millimeters. Similarly, for gas samples NaCl or KBR can be used, and gas expansions is the method. And now, some applications we will see. So, this FTIR can be used for different applications, like say analysis of pollutants in the stratosphere, ambient atmosphere, smoke and exhaust and aerosols, like say identifications of molecules, quantifications and monitoring of local and global environment and greenhouse effects, volcanic emission studies, biomass burning, screening, mapping of gas release, structure elucidations, photo oxidations, air particulate matter. So, these can also be analyzed, and methods active and passive remote sensing, multi-pass and long path gas cells, mobile and airborne FTIR spectrometers and gas chromatography, infrared spectroscopy. These are also some methods which are used, and analytes like say SOx, NOx, pHs, nitro pH and nitro aerosols, volatile organic carbons and BTs, BTX and hydrocarbons, ozone, hydrogenated compounds, HCN, H2S, O3, PCB phenols, photo dissociation and oxidation products of phenols, metabolites of terpenes and pheromones. So, these are different application, and I will give you some applications which is reported in this literature. So, here it is giving us two, this is A and this is B. So, A is nothing but FTIR spectra of GAC before and after SABA, that is simultaneously adsorption and bioaccumulation method. So, if we take say water sample and in a batch scale water samples and adsorption, then that will be only adsorption. If we use microbes and the pollutants, then also then will be biodegradations, or in case of conservative pollutants like say arsenic, those arsenic will not be degraded by the microorganisms, it will be captured and stored. So, that is bioaccumulation. And if we use simultaneously the adsorbents and microbes in the beaker, so simultaneously adsorptions and bioaccumulation will take place. Now, whether the biolayer is formed or not, that can also be confirmed by the FTIR study, as shown here. FTIR for GAC we are seeing here, but when we are taking that the GAC sample after the simultaneous adsorption bioaccumulation or SABA, so then we see that some new peaks have arisen here. So, this peak is because of the protein and lipid. So, that protein and lipid is associated with the microbial biomass. So, that way also we can correlate. So, this type of environmental applications we will find in many cases where FTIR or any other instrumental analysis can give us some insurance that yes, this phenomena is available in that particular case. So, now we will see the advantage of this FTIR spectrometry, so and its disadvantage as well. So, FTIR stands for Fourier transform infrared spectrometry, is highly sensitive and quick method to achieve high quality spectrum, and this spectroscopy gives better signal to noise ratio compared to dispersive instrument, and gases, solids as well as liquid can be analyzed by FTIR, and by using FTIR no external calibration is required, and FTIR is non destructive technique. Organic compounds and inorganic compounds can be identified, and mechanical breakdown is less compared to other instruments, because the mirror which is attached in the interferometer is the only moving point in FTIR, and FTIR can identify even small concentrations of contaminants, and it has some disadvantage that FTIR instrument have only single beam, while dispersive instrument generally have a double beam. So, these are some advantage and disadvantage. After that we have seen that the GC, HPLC, FTIR, UV visible we have discussed. Apart from that, many other instruments are also used, as I have mentioned in the previous class. So, those instruments are not that important particularly for the analysis of water quality parameters or any environmental quality parameters, but those will be very useful for studying different aspect of environmental pollution and develop technologies for the treatment of wastewater or cleaning air. So, up to this in this class, thank you very much for your patience.